Hello everyone, welcome back to Bindas Botany. Today I am going to discuss the most important MCQ on restriction enzyme or restriction endonucleases. This MCQ will help those who have prepared for TGT Biology, PGT Biology, SSB lecturer, NEET, CPET, CUVT, etc. Those who are new to my channel, please subscribe my channel. Let's begin. Question number 1. Who discovered restriction enzymes? Option A. Watson and Crick. Option B. Jacob and Monod. Option C. Natham, Arber and Smith. Option D. Boyer and Cohen. Correct answer is option C. Nathan, Arber and Smith. The next question number 2. Restriction enzyme are also called option A. Molecular knife. Option B. Molecular scissor. Option C. Molecular scalpel. Option D. All of these. Correct answer is option D, all of these. The next question number 3. Restriction enzyme are enzymes. Option A, capable of cutting DNA molecule. Option B, capable of adding nucleotide to the 3' OH end. Option C, capable of restricting protein synthesis. Option D, capable of joining DNA molecule. Correct answer is option A, capable of cutting DNA molecule. The next question number 4. The first restriction enzyme discovered was option A. EcoR1, option B. Hind2, option C. BAM-HI, option D. TAC1. Correct answer is option B. Hind2. The next question number 5. First restriction enzyme was isolated from option A. E. coli, option B. Haemophilus influenzae, option C. Pseudomonas, option D. Xanthomonas. Correct answer is option B. Haemophilus influenzae. The next question number 6. Restriction enzymes are obtained from option A. Humans, option B. Virus, option C. Bacteria, option D. Plant. The correct answer is option C. Bacteria. Haemophilus influenza is a bacteria. The next question number 7. The sequence recognized by the restriction enzyme to cut the DNA is called option A. Recognition site, option B. Restriction site, option C. Both and B, option D. Cleavage site. The correct answer is option C. Both and B. The next question number 8. Restriction enzymes option A are present in bacteria and are involved in host restriction system. Option B cleave viral DNA inside bacterium. Option C are enzymes involved in defense against bacteriophage. Option D all of these. The correct answer is option D all of these. The next question number 9. Which of the following are true regarding restriction enzyme? Option A. Restriction enzyme are used to cut DNA molecule. Option B. Restriction enzyme are used to construct restriction map. Option C. Restriction enzyme are used in RFLP. Option D. All of these. Correct answer is option D. All of these. The next question number 10. Restriction enzymes are also known as option A. Polymerase, option B. Ligase, option C. Endonucleases, option D. Exonucleases. The correct answer is option C. Endonucleases. The next question number 11. Which of the following enzyme cut DNA at a specific site? Option A. DNA polymerase, option B. DNA ligase, option C. Restriction endonuclease, option D. RNA polymerase. Correct answer is option C. Restriction endonuclease. The next question number 12. Restriction enzymes. Option A are endonucleases which cleave DNA at a specific site. Option B make DNA complementary to an existing DNA or RNA. Option C cut or joining DNA fragment. Option D are required in vectorless direct gene transfer. Correct answer is option A are endonucleases which cleave DNA at a specific site. The next question number 13. The first type 2 enzyme isolated was option A EcoR1, option B Hind2, option C BAM-HI, option D SAL1. The correct answer is option B Hind2. The next question number 14. What is the natural function of restriction enzyme in bacteria? Option A DNA replication, option B repair DNA, option C defend against viruses, option D produce protein. Correct answer is option C defend against viruses. The next question number 15. Restriction enzymes capable of making internal cut in a DNA molecule is called Option A, restriction exonuclease. Option B, restriction endonuclease. Option C, both and B. Option D, S1 nuclease. Correct answer is option B, restriction endonuclease. The next question number 16. The type of restriction enzyme used in rDNA technology is option A, type 1, option B, type 2, option C, type 3, option D, all of these. The correct answer is option B, type 2. The next question number 17. Restriction site of type 2 enzymes. Option A, generally are palindromic sequences. Option B, consist of 4 to 6 base pairs. Option C, mostly are palindrome with rotational symmetry. Option D, all of these. The correct answer is option D, all of these. The next question number 18. The term restriction in restriction enzyme refer to option A, restricting replication, option B, restricting transcription, option C, restricting phage infection, option D, restricting ligation. The correct answer is option C, restricting phage infection. The next question number 19. Eco R1 caught DNA at option A, AAGCTT, option B, GAATTC, option C, GGATCC, option D, CCGG. Correct answer is option B, GAATTC. GAATTC. The next question number 20. Eco R1 always caught DNA molecule at a particular point by recognizing a specific sequence between option A, G and A, option B, T and C, option C, A and A, option D, T and T. Correct answer is option A, G and A. The next question number 21. The recognition site of eco R1 is option A 5 prime GAATTC 3 prime, option B 5 prime 2 GTCGSC 3 prime, option C 5 prime and 2 AAGCTT2 3 prime and option D 5 prime GGATCC2 3 prime and. The correct answer is option A GAATTC GAATTC from 5 prime end to 3 prime end. The next question number 22. Sticky ends are produced by option A, DNA polymerase, option B, ligase, option C, blunt end cutter, option D, set and restriction enzymes. Correct answer is option B, set and restriction enzymes. The next question number 23. Which type of enzyme caught both strand of DNA at a specific point? Option A, exonuclease, option B, endonuclease, option C, ligase, option D, helicase. Correct answer is option B, endonuclease. The next question number 24. How many type of restriction enzyme are there? Option A, 2 type, option B, 3 type, option C, 4 type, option D, 5 type. The correct answer is option C, 4 type. These are type 1, type 2, type 3 and type 4. The next question number 25. Which type of restriction enzyme is widely used in molecular biology? Option A type 1, option B type 2, option C type 3, option D type 4. Correct answer is option B type 2. The next question number 26. Which enzyme does not require ATP for cleavage? Option A type 1, option B type 3, option C type 2, option D all require ATP. The correct answer is option C type 2. The next question number 27. Hind2 caught a DNA molecule at a specific point by recognizing a specific sequence of option A 2 base pair, option C 3 base pairs, option C 5 base pairs, option D 6 base pair. The correct answer is option D 6 base pairs. The sequence is GTCGAC. The next question number 28. Hind2 is a option A polynucleotide, option B polypeptide, option C polysaccharide, option D bacterium. The correct answer is option B polypeptide. The next question number 29. The end generated by Hind2 are option A blunt end, option B sticky end, option C sealed, option D phosphorylated. The correct answer is option A blunt end. Hind2 caught the DNA between C and G and the end formed is blunt end. The next question number 30. The enzyme Hind3 cut DNA between option A and A, option B, A and G, option C, G and A, option D, T and A. The correct answer is option C, G and A. The next question number 31. The end generated by Hind3 are option A, blunt end, option B, sticky end, option C, sealed, option D, phosphorylated. The correct answer is option B, sticky end. This is a sticky end. The next question number 32. A DNA fragment with blunt end can be joined by using option A, ligase, option B, polymerase, option C, helicase, option D, restriction enzyme. The correct answer is option A, ligase or DNA ligase. The next question number 33. What is the full form of EcoR1? Option A, Escherichia coli restriction 1. Option B, enzyme colony restriction 1. Option C, eukaryotic coli restriction. Option D, Escherichia coli RY13. The correct answer is option A, Escherichia coli restriction 1. The next question number 34. Eco R1 is obtained from option A, Escherichia coli R13, option B, Escherichia coli RY13, option C, Escherichia coli R225, option D, Escherichia coli RC. The correct answer is option B, Escherichia coli RY13. The next question number 35. The first letter in the name of restriction enzyme comes from option A, species, option B, genus, option C, strain, option D, order. Correct answer is option B, genus. The next question number 36. The letter R in eco R1 is derived from option A, the name of a genus, option B, the name of strain, option C, the name of species, option D, none of the above. Correct answer is option B, the name of the strain. The next question number 37. The R in eco R1 represent option A, restriction, option B, strain RY13, option C, replication, option D, recombinant. Correct answer is option B, strain RY13. The next question number 38. What does the 1 in eco R1 represent? Option A, fast enzyme isolated, option B, fast gene edited, option C, fast protein sequenced, option D, fast endonuclease in lab. The correct answer is option 1, fast enzyme isolated. The next question number 39. DNA methylation prevent restriction by option A, blocking the active site, option B, modifying recognition site, option C, inhibiting binding, option D, inhibiting transcription. The correct answer is option B, modifying recognition site. The next question number 40. Isosomers recognize option A, different sequences, option B, the same sequence, option C, no sequence, option D, RNA only. The correct answer is option B, the same sequences. The next question number 41. Which restriction enzyme is a neosomer of eco R1? Option A, HPA2, option B, MBO1, option C, MFE1, option D, XVAL. The correct answer is option C, MFE1. What is a neosomes? A neosomes is a restriction enzyme that recognizes the same DNA nucleotide sequences as another restriction enzyme but cleave it at a different position. The next question number 42. Restriction enzymes are used in option A, DNA fingerprinting, option B, PCR, option C, RNA splicing, option D, protein synthesis. The correct answer is option A, DNA fingerprinting. The next question number 43. Restriction endonucleases in DNA fingerprinting carry out the following processes. Option A, fragmentation of DNA. Option B, getting copies of DNA. Option C, loading DNA on agarose plate. Option D, synthesis of DNA. The correct answer is option A, fragmentation of DNA. The next question number 44. In RFLP technique, which enzyme is used? Option A, helicase. Option B, DNA ligase. Option C, restriction enzyme. Option D, DNA polymerase. Correct answer is option C, restriction enzyme. The next question number 45. Which of the following technique does not require restriction enzyme? Option A Saturn blotting, option B DNA sequencing, option C PCR, option D DNA fingerprinting. The correct answer is option C PCR, polymerase chain reaction. The next question number 46. Restriction mapping is used to option A determine gene expression, option B determine protein structure, option C locate restriction site, option D amplify DNA. The correct answer is option C locate restriction site. The next question number 47. Which of these tools is essential in recombinant DNA technology? Option A DNA polymerase, option B helicase, option C restriction enzyme, option D guidase. The correct answer is option C restriction enzyme. The next question number 48. The vectors are cut using option A polymerase, option B ligase, option C restriction enzyme, option D endopolymerase. The correct answer is option C restriction enzyme. The next question number 49. For gene cloning, both vector and insert DNA should be cut by option A same restriction enzyme, option B different enzyme, option C ligase, option D polymerase. Correct answer is option A same restriction enzyme. The next question number 50. Which of the following statement about restriction enzyme is incorrect? Option A. They are site specific. Option B. They cut RNA molecule. Option C. They are found in bacteria. Option D. They recognize palindromic sequences. Correct answer is option B. They cut RNA molecule, which is incorrect. The next question number 51. Sticky ends help in option A. Destroying DNA. Option B. Methylation. Option C. Easy ligation. Option D. DNA replication. Correct answer is option C. Easy ligation. The next question number 52. Blunt end are option A. Easy to ligate. Option B. Difficult to ligate. Option C. Auto-ligated. Option D. Self-destructive. The correct answer is option B. Difficult to ligate. This is a sticky end, and this is a blunt end. Blunt end are difficult to ligate or difficult to join, and sticky end are help in easy ligation or easy to join. The next question of 52. What protect bacterial DNA from its own restriction enzyme? Option A ligase, option B methylation, option C sticky ends, option D helicase. Correct answer is option B methylation. The next question number 54. Which of the following has a blunt end? Option A echo R1, option B hind 3, option C SMA1, option D BAM H1. Correct answer is option C SMA1. The recognition site of SMA1 is CCCGGG. The next question number 55. Which of the following restriction enzymes produce blunt end? Option A SMA1, option B HAE3, option C LU1, option D all of these. Correct answer is option D all of these. The next question number 56. Which of the following restriction enzyme produce blunt end? Option A SAL1, option B EcoR5, option C XHO1, option D HIND3. Correct answer is option B EcoR5. The recognition site of EcoR5 is GATATC. It got blunt end. The next question number 57. Blunt end are formed by option A EcoR1, option B SMA1, option C HIND3, option D BAM H1. Correct answer is option B SMA1. The next question number 58. Which restriction enzyme produce a 5' overhang? Option A BAM H1, option B SMA1, option C ALU1, option D HAE3. Correct answer is option A BAM H1. The recognition site of BAM H1 is GGATCC. The next question number 59. The overhang created by echo R1 is option A 5', option C 3', option C blunt, option D none. The correct answer is Option A 5 prime. The next question number 60. Enzyme that leave no overhang. Option A sticky end cutter. Option B blunt end cutters. Option C ligase. Option D helicase. Correct answer is option B blunt ends. The next question number 61. Palindromic sequences are generally how long? Option A 3 to 4 base pair. Option B 4 to 8 base pair. Option C 8 to 12 base pair. Option D 1 to 2 base pair. Correct answer is option B 4 to 8 base pair. The next question number 62. Which is not a palindromic sequence? Option A GAATTC. Option B AAGCTT. Option C GTCAAC. Option D GATATC. The correct answer is option C GTCAAC. The next question number 63. Which one of the following represent a palindromic sequence in DNA? Option A 5 prime CATTAG to 3 prime, and the other strand is 3 prime GATAC to 5 prime, and option B 5 prime and GATACC to 3 prime, and other is 3 prime CCTAAG to 5 prime, and option C 5 prime GAATTC to 3 prime, and the next strand is 3 prime CTTAAG to 5 prime, and the next option D 5 prime CCAATG to 3 prime, and its complement strand is 3 prime GAATCC to 5 prime. The correct answer is option C 5 prime GAATTC to 3 prime, and its complement strand 3 prime CTTAAG to 5 prime. The next question number 64. Which of the following is an application of restriction enzyme? Option A gene silencing, option B gene editing, option C protein folding, option D mRNA splicing. Correct answer is option B gene editing. The next question number 65. Restriction enzyme can be inactivated by option A cooling, option B heating, option C ligase, option D DNA. Correct answer is option B heating. The next question number 66. DNA fragment created by restriction enzyme can be separated using option A ELISA, option B Western blot, option C gel electrophoresis, option D spectrophotometer. Correct answer is option C gel electrophoresis. The next question number 67. Which pair of enzyme is commonly used for cloning DNA fragment? Option A ECO R1 and BAM H1, option B DNA polymerase and ligase, option C Taq polymerase and helicase, option D SMA1 and primase. Correct answer is option A ECO R1 and BAM H1. The next question number 68. The term iso Sejomer refer to enzyme that option A have the same source, option B recognize the same sequence, option C cut at different location, option D require ATP. The correct answer is option B recognize the same sequence. The next question number 69. Neosegomers are enzyme that option A recognize the same sequence but cut differently, option B recognize the different sequence, option C only work in acidic pH, option D denatured protein. The correct answer is option A recognize the same sequence but cut a different place. The next question number 70. Restriction enzyme always cut option A RNA, option B DNA randomly, option C at specific recognition sequence, option D only mitochondrial DNA. Correct answer is option C at specific recognition sequences. The next question number 71. The bacterial strain used to derive ECO R1 is option A Thermos aquaticus, option B E. coli RY13, option C Streptococcus pyogenes, option D Bacillus subtilis. Correct answer is option B E. coli RY13. The next question number 72. Type 2 restriction enzyme require option A ATP, option B NAD+, option C no cofactor, option D magnesium ion. Correct answer is option D magnesium ion. The next question number 73. Methylation of adenine or cytosine bases in bacteria. Option A. Prevent transcription. Option B. Promote mutation. Option C. Protect DNA from endonuclease. Option D. Enhances replication. Correct answer is option C. Protect DNA from endonuclease. Here the methylation of cytosine occur at 5 carbon atom. The next question number 74. Which enzyme leave a 3' overhang? Option A eco R1, option B PST1, option C SMA1, option D ALU1. Correct answer is option B PST1. The recognition site is CTGCAG. It forms sticky end. The next question number 75. Enzyme that cut DNA at random position are option A type 1 RE, option B type 2 RE, option C DNA polymerase, option D helicase. Correct answer is option A type 1 restriction endonuclease. The next question number 76. Enzyme PBU2 recognize option A GAATTC, option B CAGCTG, option C GATATC, option D CCTAGG. Correct answer is option B CAGCTG. The recognition site is CAGCTG, which cut between G and C and form blunt end. The next question number 77. Which of the following is not a type 2 restriction enzyme? Option A Hind3, option B EcoR1, option C BAMH1, option D EcoK1. Option D is the correct answer, EcoK1. The next question number 78. Which enzyme produce blunt end? Option A ALU1, option B Hind3, option C EcoR1, option D BAMH1. Correct answer is option A ALU1. The next question number 79. Type 1 enzymes differ from type 2 by option A having no recognition site, option B requiring ATP and cutting away from site, option C producing sticky end only, option D only cutting RNA. Correct answer is option B requiring ATP and cutting away from site. The next question number 80. RM system in bacteria stands for option A replication mutation, option B restriction modification, option C recombination mutation, option D repair mutation. Correct answer is option B restriction modification. The next question number 81. Type 3 restriction enzyme require option A ligase, option B ATP and recognition site, option C RNA template, option D heat. Correct answer is option B ATP and recognition site. The next question number 82. Which of the following is true about type 2 restriction enzyme? Option A. Recognize sequence and cut at a distant site. Option B. Require ATP. Option C. Cut at a recognition site. Option D. Require DNA polymerase. Correct answer is option C. Cut at a recognition site. The next question number 83. Which enzyme is used to generate DNA fingerprint? Option A. EcoR1. Option B. Ligase. Option C. Polymerase. Option D. Primase. Correct answer is option A, EcoR1. The next question number 84. The number of cut in DNA by restriction enzyme depend on option A, number of enzymes, option B, size of genome, option C, number of recognition site, option D, temperature only. Correct answer is option C, number of recognition site. The next question number 85. Which of the following is not used in restriction digestion? Option A DNA, option B restriction buffer, option C DNA ligase, option D restriction enzyme. Correct answer is option C DNA ligase. The next question number 86. A 6 base pair recognition site occur once in option A 64 base pair, option B 4096 base pair, option C 1024 base pair, option D 256 base pair. Correct answer is option B 4096 base pair. The next question number 87. Which statement is false? Option A. Restriction enzyme cut DNA at a specific site. Option B. Sticky and enhanced ligation. Option C. Restriction enzyme are present in eukaryote. Option D. Restriction may show cut sides. Correct answer is option C. Restriction enzymes are present in eukaryotes. The next question number 88. Which enzyme is used in gene editing along with guide RNA? Option A, EcoR1. Option B, CRISPR-Cas9. Option C, Taq polymerase. Option D, ligase. Correct answer is option B, CRISPR-Cas9. The next question number 89. A DNA molecule has three EcoR1 sites. How many fragment are how many fragment after digestion? Option A 2, option B 3, option C 4, option D 5. Correct answer is option C 4. The next question number 90. Which of the following is not a role of restriction enzyme? Option A DNA cleavage, option B cloning, option C replication, option D mapping. Correct answer is option C replication. The next question number 91. Why are sticky ends preferred in cloning? Option A reduce ligation, option B increase stability, option C increase pairing specificity, option D help denaturation. Correct answer is option C increase pairing specificity. The next question number 92. Which of the following does not influence restriction enzyme activity? Option A temperature, option B pH, option C salt, option D light. Correct answer is option D light. The next question number 93. Type 2 restriction enzyme differ from type 1 in that they option A are ATP dependent, option B cut randomly, option C cut at recognition site, option D cut only single strand. Correct answer is option C cut at recognition site. The next question number 94. BAM H1 generated option A 3' overhang, option B 5' overhang, option C blunt end, option D both end B. Correct answer is option B 5' overhang. The next question number 95. An enzyme cutting at 5' CCCGGG to 3' end is option A Eco R1, option B BAM H1, option C SMA1, option D Hind 3. Correct answer is option C SMA1. The next question number 96. Restriction endonuclease cut option A sugar phosphate backbone, option B nitrogen base, option C histone protein, option D RNA. Correct answer is option A sugar phosphate backbone. The next question number 97. Restriction endonucleases are useful in option A breaking DNA at a specific site, option B creating sticky ends, option C both A and B, option D crossing over. Correct answer is option C both A and B. The next question number 98. Restriction endonucleases are used in genetic engineering to form option A recombinant molecule of protein, option B recombinant molecule of DNA, option C recombinant molecule of protein and DNA, option D recombinant cell. Correct answer is option B recombinant molecule of DNA. The next question number 99. Restriction endonucleases are enzymes which option A make cut at specific positions within the DNA molecule. Option B recognize a specific nucleotide sequence for binding of DNA ligase. Option C restrict the action of the enzyme DNA polymerase. Option D remove nucleotide from the ends of the DNA molecule. Correct answer is option A make a cut at specific positions within the DNA molecule. The next question number 100. The usual source of restriction endonucleases used in gene cloning is option A fungi, option B bacteria, option C plant, option D viruses. Correct answer is option B bacteria. The next question number 101. Restriction endonucleases are option A synthesized by bacteria as part of their defense mechanism, option B used in vitro DNA synthesis, option C present in mammalian cells to degrade DNA when the cell die. Option D used in genetic engineering for ligation of two DNA molecules. Correct answer is option A synthesized by bacteria as part of the defense mechanism. The next question number 102. Restriction endonuclease is employed for cutting option A single stranded DNA, option B double stranded DNA, option C RNA fragment, option D mRNA. Correct answer is option B double stranded DNA. The next question number 103. Endonuclease is employed in option A transcription, option B translation, option C genetic engineering, option D DNA replication. Correct answer is option C genetic engineering. The next question number 104. Restriction endonuclease used widely in RDT are obtained from option A plasmid, option B bacterial cell, option C bacteriophage, option D all prokaryotic cell. Correct answer is option B bacterial cell. The next question number 105. Restriction endonucleases are enzymes that option A restrict the action of other enzyme, option B break phosphodiester bond between the specific nucleotide of DNA, option C break nucleus into pieces, option D break hydrogen bond between two strands of DNA. Correct answer is option B break phosphodiester bond between specific nucleotide of DNA. The next question number 106. Which of the following statement is not correct regarding EcoR1 restriction endonuclease enzyme? Option A. It is isolated from Escherichia coli RY13. Option B. Its recognition sequence is 5' GAATTC to 3' end and 3' CTTAAG to 5' end. Option C. It produces complementary blunt ends. Option D. None of the above. Correct answer is option C. It produces complementary blunt ends. The next question number 107. Term restriction in restriction endonuclease enzyme refer to option A. Cleaving of a phosphodiester bond in DNA by the enzyme. Option B. Cutting of DNA at specific position only. Option C. Preventing of bacteriophage multiplication in bacteria. Option D. Cutting each of two strands of DNA at specific point in sugar phosphate backbone. Correct answer is option C. Preventing of bacteriophage multiplication in bacteria. The next question number 108. Function of restriction endonuclease enzyme is option A. It is useful in genetic engineering. Option B. It protects the bacterial DNA against foreign DNA. Option C. It is helpful in transcription. Option D. It is helpful in protein synthesis. Correct answer is option B. It protects the bacterial DNA against foreign DNA. The next question number 109. Type 2 restriction enzymes. Option A. Recognize the specific sequence and cleave DNA at that specific sequence. Option B. Do not recognize or cleave the specific sequence. Option C. Recognize specific sequence but cleave non-specific sequence. Option D. None of the above. Correct answer is option A. Recognize the specific sequence and cleave DNA at that specific sequence. The next question number 110. Cleavage style of restriction enzymes include option A blunt end style, option B sticky end style, option C no style, option D both and B. Correct answer is option D both and B. The next question number 111. The substrate for restriction enzyme is option A single stranded RNA, option B protein, option C double stranded DNA, option D single stranded DNA. Correct answer is option C double stranded DNA. The next question number 112. Identify the statement which is not true for restriction enzymes. Option A. Some 900 restriction enzymes have been isolated till date. Option B. They cut each of the two strands of DNA at specific point through their sugar phosphate backbone. Option C. The naming of restriction enzymes follows the sequence strain name followed by specific species name followed by genus name. Option D. The restriction enzyme can be both exonuclease as well as endonuclease. Correct answer is option C. The naming of restriction enzyme follows the sequence strain name followed by species name followed by genus name, which is not true. The next question number 113. The natural function of a restriction enzyme is to option A. Cut foreign DNA. Option B. Remove intron from the RNA transcript. Option C. Remove exon from the RNA transcript. Option D. Facilitate mRNA formation from nucleotide. Correct answer is option A. Cutting foreign DNA. The next question number 114. The source of the restriction enzyme hint to is option A. Escherichia coli RY13. Option B. Haemophilus influenzae RD. Option C. Bacillus amyloidosis. Option D. Streptomyces albus. Correct answer is option B. Haemophilus influenzae RD. The next question number 115. Restriction enzymes are option A. Not always required in genetic engineering. Option B. Essential tool in genetic engineering. Option C. Nuclease that cleave DNA at a specific site. Option D. Both B and C. Correct answer is option D. Both B and C. The next question number 116. Which of the following is not a restriction endonuclease? Option A. EcoR1. Option B. Hind3. Option C. PST1. Option D. BNH1. Correct answer is option D. BNH1. The next question number 117. Which of the following is a restriction endonuclease? Option A. protease 1, option B DNase 1, option C RNase, option D Hind 2. Correct answer is option D Hind 2. The next question number 118. In PBR 322, tetracycline resistance gene has recognition site for which of the following restriction endonuclease? Option A Hind3, option B BAMH1, option C EcoR1, option D PST1. Correct answer is option B BAMH1. The next question number 119. In order to insert alien DNA at the site of tetracycline resistance gene in PBR 322, the restriction endonuclease used can be option A PV1, option B CLA1, option C SAL1, option D PST1. Correct answer is option C SAL1. The next question number 120. Which of the following restriction endonuclease cannot be used while using PBR 322 as a cloning vector? Option A PVU2, option C BAMH1, option C SAL1, option D PST1. Correct answer is option A PVU2. The next question number 121. Single standard unpaired extensions formed by restriction enzyme upon cleavage is called as option A blunt end, option B flash end, option C sticky end, option D none of these. Correct answer is option C sticky ends. The next question number 122. Restriction enzymes option A are present in bacteria and are involved in a host restriction system. Option B cleavage viral DNA inside bacterium. Option C are enzyme involved in defense against bacteriophages. Option D all of these. Correct answer is option D all of these. The next question number 123. For cloning, restriction enzyme with sticky ends are used for option A easy of transformation, option B easy insertion into plasmid of DNA segment from defense sources, option C easy identification of plasmid with antibiotic resistance, option D easy identification of plasmid having inserts. Correct answer is option B easy insertion into plasmid of DNA segment from different sources. The next question number 124. The restriction enzyme bind to and cut the sequence of DNA which usually are option A antiparallel, option B not symmetrical about the midpoint, option C symmetrical about the midpoint, option D random sequences. Correct answer is option C symmetrical about the midpoint. The next question number 125. In bacteria, the restriction phenomena occurs naturally as option A bacteria produce enzymes, option B for survival, option C for efficient cloning, option D destruction of DNA of the bacterium. Correct answer is option A. Bacterial produce enzymes. The next question number 126. HAE3 and ALU1 are restriction enzyme that cut straight across the double helix producing option A sticky end, option B blunt end, option C codon, option D anticodon. Correct answer is option B blunt ends. The next question number 127. Zinc finger nucleases is option A natural restriction enzyme, option B type of polymerase enzyme, option C artificial restriction enzyme, option D type of helicase enzyme. Correct answer is option C artificial restriction enzyme. The next question number 128. Which of the following statement are true regarding restriction enzyme? Option A type 1 and type 3 enzymes cut far away from the restriction site. Option B type 2 cut DNA within a restriction site. Option D EcoR1 is a type 2 restriction enzyme. Option D all of these. Correct answer is option D all of these. The next question number 129. Which of the following is the most important discovery that led to the development of recombinant DNA technology? Option A discovery of double helix model by Watson and Crick. Option B discovery of DNA as genetic material. Option C discovery of restriction enzyme. Option D all of these. Correct answer is option C discovery of restriction enzyme. The next question, last question, question number 130. Restriction enzyme recognize option A tRNA sequence, option B repetitive DNA, option C palindromic sequences, option D only AT regions. Correct answer is option C palindromic sequences. Here these are the type 2 restriction enzymes and with source and recognition sequence and fragments you have to remember. These all are type 2 restriction enzymes. Thank you for watching this video. Please like, share, comment and subscribe my channel.